Hello, good evening. Hello, good evening. You can hear me, eh? You can hear me clearly today, right? So, how are you, people? How are you? How are you? How are you? Komada, doing good. Right, you can hear me, eh? Please send me that message. Can you send it to the main chat line? No, I'll open the main chat line first, and no one, everyone. Now you can send messages to everyone, right, to the main chat line. Send your messages to the main chat line, people, so I can see it clearly. Can you? Right, send your messages to Fine. Nick. Good evening. Good evening. I'm fine. Who's this? Right, Rishma. Yes, Nick. Hari. So you have to do a small favor for me. From where do we have to start today, people? We'll start with the. We'll start with. We'll start with. Uh, we'll start with the cell structure and the function part first. Neil. We'll start with the cell structure function lesson. So we have some questions to cover here. How many questions? I think we left the exam questions. Lama, you have to send the your replies to the main chat for everybody to see. Okay. So could you please do that? People, how many questions we have to discuss? How many questions remain to discuss? As I remember, we have to go from page number 19. Is it page number 19? Page 19, please. Please send me a reply. Lamai. When the main chat, main chat. Can you? Can't you send messages to the main chat? We can hear you. Okay, fine. Yes. Lovely. Good. So send me from where we have to start today. Number, page number. Come on. Come on. Come on. Can you send it? Can't you use the main chat? Can't you use the main chat? The page number 21, Lou. Can't, who's this? Okay. Mohammed Hari. Mohammed 22, ne? The Hari. So others, now I can see his chat clearly. Can others chat with everyone? Say hi. Say hi. Hi. Just say hi. Right. Page number 21. Okay, so that means we finish all the MCQs, right? So I'll consider that we completed all MCQs. We have to start from 22. 22, is that 22? 2010 A-level paper question. Am I correct? 2010 A-level paper question. 25. 25. 25, 25, 22, 22 here. 22, this one. Is it? 22. All right. 2013, yeah, 22. Am I correct, people? We'll start from 2023 paper. We'll start 2023 paper. We'll start from 2013 paper. All right, 2013 paper. Uh, 2013. Uh, 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 what is um, people? I have a small confusion about the page numbers. 22 page, mine 22. Mine 22 is slightly different. This is my 22. This is my 22. Um, find that. People, is it okay? This one 2010. Is it? Do we have to start from 2010? Please, Mohammed. Do we have to start from 2010 ALL paper question? Yes or no, please? Come on. Do we have to start from 2010? 2004, ne? 2004 ALL paper. 2004. Page number 21. Okay. Page 21. 2004. Hurry. Then 2004. Stop. Okay, because 2004. This doesn't work. 2010. Right. We'll start from there. 2004. We start. Like, okay, now. 2004. Right. I have some technical problem now. Right. 2004 A-level paper question. All right. No, it's not working. So we'll go in this setup, and with this camera, we'll try to discuss. 2004. People, what are four major concepts, including cell theory? That's the question. Now we learn only three concepts in cell theory, not four. Three concepts now. Cell is a basic. All organisms are made up of 1 ohm new cells from. Okay, thank you. All organisms are made of 1 ohm many cells. Second one, cell is a basic structural unit and the functional unit. New cells always formed by the division of the previously existing cells. Those 3 points you have to write here. Those 3 points you have to write here. Okay. Right. Question number 4. Next question. Question number 4. List 5 organelles found in eukaryotic cell and indicate the presence or absence of protein, lipid, RNA, DNA, or absence, plus sign for the presence and negative sign for the absence of the relevant column, and one function of each organelle. So turn to next page. So shall we take the organelle first one? Shall we take the organelle first one? What are you going to take first? Let's take people nucleus. Nucleus, take as the organelle. So we are discussing this tutor, which I have distributed in the class, right? Cell structure function tutor. So let's take organelle as the nucleus. Nucleus is the organelle. And then what are you going to write, people? What are you going to write? Proteins present. Proteins present. Protein present. Second one, protein present. And then lipids present. Lipids present in the plasma, what do you call the nuclear knob, right? Lipids present. Right, try to fix it. It's not responding. Right. Right. Organelles. So you can take nucleus. Nucleus. So you can take nucleus. You can take nucleus. You can take proteins present, lipids present, RNA, DNA present. Function. What are you going to write, people? Function. You can see synthesis of ribosomes, store genetic information, so on. Right. Next one you can take mitochondria. Right. Mitochondria, proteins present, lipids present, RNA present, DNA present, and then you can take function. What are you going to take? Cellular respiration, aerobic respiration, maybe aerobic respiration, or maybe helps in involves in photo respiration, right? Aerobic respiration, will take it like that. Then third one, people will take chloroplast. So proteins present, lipids present, RNA present, DNA present, right? And then functions, photosynthesis. That's correct. And then let's take ribosome as the next one. Ribosome, protein present, lipid absent, RNA present, DNA absent, function protein synthesis. You tell me another organelle, people. Golgi. I will take Golgi as the last one. Protein present, lipid present, RNA absent, DNA absent, function. What are you going to write? Formation of lysosomes. Right. So you can write that. Question 2010 A-level paper question. Name A and B in the electron micrograph diagram shown below. So you have to find out A and B. You have to find out A and B. You have to find out A and B. A and B. Right. A and B. What is A, people? You can take rough endoplasmic reticulum. A, rough endoplasmic reticulum. B, you can take as people smooth endoplasmic reticulum. Sorry, B, Golgi apparatus. Sorry, B is Golgi apparatus. B, Golgi apparatus. B, Golgi apparatus. Right? B, Golgi apparatus. And then state two functions of each of A and B. Two functions of A and B. Okay, you can take RER function. What we said, people? Involving in synthesis of glycoproteins. What else? Formation of transport vesicles and all. B, Golgi complex will take one function here near lysosome production. What else we said, people? The involving in packaging near we said something, packaging, distribution, what we said for Golgi, involving in collecting, modifying, packaging, and distribution of materials. Right. Next one, people. Third one, what is cytoskeleton? What is cytoskeleton? You can see that the supporting structure of the cell, which maintains the shape. Cytoskeleton is a supporting structure of the cell which maintains the shape. You can further say that, made up of microtubules and actin filaments, or microfilaments and intermediate filaments. So that is the cytoskeleton. And then state three functions of cytoskeleton. Cytoskeleton, three functions we can say: provide strength to the cytoplasm, encourage of organelles and cytosolic enzymes, and then movement of cytoplasm, cytoplasmic streaming, and also you can say people maintain shape of the cell, maintain shape of the cell. Then next question, endosymbiosis theory, people will we don't discuss now this endosymbiosis theory. It's not in our syllabus. It explains basically the formation of mitochondria and chloroplast in eukaryotic cells, right? They are actually some of the bacteria, right? Early bacteria form some symbiosis with an eukaryotic cell, right? That is what you call endosymbiotic theory. We'll skip that question, okay? Just cut with one line. It's out of syllabus. Question 2013 old syllabus. Given below is a line diagram which shows the ultrastructure of the mitochondrion, right? So this question, right? Ultrastructure of the mitochondrion. Label A, B, C, D, right? Label A, B, C, D. What is A? A, outer membrane. B, I'm taking as inner membrane. C, cristae. D, what is D? Mitochondrial matrix. Mitochondrial matrix. Right, that's good. Right, 2014 A-level paper question. 2014 A-level paper question. Name two organelles in living cells which are involved in detoxification of some substances produced during metabolism. Detoxification. What are the organelles involving in detoxification, Lamai? Two organelles only. One is SCR. What is the other one? Other one is peroxisome. SCR and peroxisome. Moving to next page. 2018 A-level paper question. State three concepts of cell theory. We discussed that. So I'm going to skip it. And then what are the internal structural features of eukaryotic cells that are different from those of the prokaryotic cells? What are the internal structural features of eukaryotic cells? What are you going to write? Can you tell me? Yeah, the question, what are the internal structural features of eukaryotic that are different from that of the prokaryotes? You can say people membrane-bounded organelles. Prokaryotic cells, the membrane-bounded structures absent. We'll say that people No membrane-bounded organelles. Membrane-bounded organelles and other subcellular components are present. And then you can write about the nucleus. What are you going to say, people? DNA, organized nucleus present, organized nucleus absent. Or else you can say that the DNA is circular and then linear DNA, right? And then you can say people 70S ribosomes present in prokaryotes, both 70S and 80S ribosomes available. And then internally, so don't write about the cell wall, don't write about cell wall. And yeah, those features I think are sufficient as the answer for this question. Question part 3. A student was provided with an epidermal peel of onion mounted on a glass slide and a light microscope. State the correct sequence of steps that should be followed to observe shape of onion epidermal cells. Onion epidermal cells under the light microscope. Provided with an epidermal peel onion mounted on the glass slide and light microscope. Correct sequence of the steps that should be followed to observe the shape of the onion. I think so. You don't have to talk about the preparation. Remove epidermal peels, drop it in water, transfer with the ethically unknown. Because they say that peels in onion mounted on the glass slide. So what you have to write now is the procedure to observe, right? Procedure to observe this epidermal peel. So what are you going to say, people? Turn on the low power, look through the microscope, and then place the slide containing the epidermal peel, adjust the mirror, and then use the coarse focus control, and then use fine focus control to get the image. Use the coarse focus control and the fine focus control, and then get the image Okay, so that's the procedure. Now the resource book people, the practical part, they have given certain steps. So you have to follow those steps and then complete the answer. 2019 annual paper state the function of rough endoplasmic reticulum. Rough endoplasmic reticulum functions they ask: transport protein synthesized by bound ribosomes. Then you can see people synthesizing of glycoprotein, and then production of transport vesicle, synthesized membrane phospholipids, then facilitate growth of the own membrane by adding phospholipids, proteins, and carbohydrates. Those points you have to write here. Question 2019, fourth one. Name 3 types of vacuoles seen in organisms. In our syllabus, they have mentioned 3 types of vacuoles: central vacuole, food vacuole, and the contractile vacuole. Right, 2020 A-level paper question. Name the structure that help in cytoplasmic streaming and movement of chromosomes, and state the structural component. 2020, name the structure that helps in cytoplasmic streaming. So we say that people, cytoplasmic streaming is cytoskeleton. So answer has to be cytoskeleton. Now in the early syllabus, people, they have taken the component, they have mentioned which component, whether it is the microtubule, intermediate filament, or the microfilaments. Exactly, they have mentioned which component participate in it, right? But now you don't need to know that. Just take the answer as the cytoskeleton, that is enough. Structural component, microtubules, microfilaments, and intermediate filaments. Next one, state the location of the secondary cell wall of a plant cell and name substance present in it other than cellulose. Name the location of the secondary cell wall. Now, how do you describe the location of the secondary cell wall present between plasma membrane and primary cell wall? And then and uh, and name the substance present in it other than cellulose, lignin and suberin. Yeah. Right, 2022 A-level paper question. What is a cell junction? What is a cell junction? Structure at which cytoplasm of adjoining cells are joined, right? Is the cell junction. Name cell junctions that carries out each of the following. Prevent leakage of extracellular fluid. Tight junction, that's correct. Tight junctions, very good. Allowing exchange of materials between cells, that is going to be gap junctions, correct, right? And then next people, 2024 A-level paper question. 2024 A-level paper question. Name the protein with alpha helix structure, which is a component of intermediate filaments of animal cytoskeleton. Name a protein with alpha helix structure. Name a protein with alpha helix structure which is component of intermediate filament. What is that, people? Keratin. That's correct. Name component present in the cell wall of both bacteria and cyanobacteria, but not in the cell wall of archaea. That is peptidoglycan. Your answers are correct. Very good. And then, people, what act as the object for the eyepiece lens when the specimen is observed through the compound light microscope? What is becoming object for the eyepiece lens? Neither. The image formed by the objective lens. Image formed by objective lens. Right. Question part B. What is used to stain specimen for observation through transmission electron microscope? Transmission electron microscope, we say heavy metals. If it is scanning electron microscope, what we say then? If it is scanning electron microscope, what do you say there? Huh? Gold. Hurry. You look at the state two functions carried out by both rough and smooth endoplasmic reticulum, and two functions carried out only by the smooth endoplasmic reticulum. So it says both. I'm taking synthesis of phospholipids, production of transport vesicles, right? Those two. And then the next one, by SCR only. What are you going to write for that? SCR only. SCR only. Detoxification. That is correct. And synthesis of oil, steroids, storage of calcium, metabolism of carbohydrates. Metabolism of carbohydrates. Right? Next one, people. Next one. Name the most abundant glycoprotein in the extracellular matrix of animal cells. Name the most abundant glycoprotein in extracellular matrix. The most abundant glycoprotein in extracellular matrix of animal cells. What is that? Collagen. Good. Name the subcellular component that carries out each of the following activities in the cell. Transport residue materials out of the cell. Lysosome. Very good. And then cytoplasmic streaming. Cytoskeleton. Very good. I have a very nice set of students. Maybe I don't have to sell the answer, right? They are typing it. Right. Good. And then people, which organelle in plant cells involving in formation of the cell plate? Which organelle during, okay. Which organelle involving in formation of the cell plate? Can you tell me the answer for that? Golgi apparatus. That's good. Okay, part B, neither. Part B is which organelle during cytokinesis? I think this question is little unclear. That's all for the dispute. That's all for dispute. Then, oh, cell cycle tute. Can you take cell cycle tute? I think I left few questions in the last class. I said that I'm going to discuss it online with you, right? And someone tell me the page number. Today, people, I have my smart board is stuck at the moment. I don't know the reason. I had to find out. But I did certain improvements here, right? Now the cameras are clear, ne? Not like I think last time. Cameras are clear, not gloomy also. Ne? It was little darker in the studio because of the camera settings. Now we improved it. Hope yeah, you can see me clearly. Ne? Okay. So, people, what is page number 17? Page number 17. 17, no question number. Ne? Question 17, ne? No page numbers. Question. No page numbers. Hari, you tell me which question. We did 13, ne? 13 we did. Clear. Question number 15. 14 we did. Then we have to do 15. The diagram present, okay. Right. Diagram present in a plant cell in which the diploid number of chromosome is 6. Okay. So this is the diagram given. Right. This diagram is given. This diagram is given. This is the nucleus. That's the nucleus. That is the nucleus. This is the nucleus. And then we have, these are the chromosomes. These are chromosomes. Okay? So you can see the homologous, they have the same shape. Homologous ones are having the same shape. All right. Homologous, they have same shape. So must do B part in 14th question also. B part in 14th question. We'll come that later, okay? We'll come back for that later. Draw a diagram to show this cell during anaphase of mitosis. This cell during the anaphase of mitosis, right? Now, we'll draw first, people, and without going into anaphase, we'll draw first this cell after passing G2. Let's draw the diagram, people, after passing S phase when it comes to G2, right? So I'll draw it here. What happened during the S phase? This is going to replicate, right? This is also going to replicate and then form sister chromatids, right? This one going to be like that. This one going to be like that. This one going to be like this. This one is also now forming partner. That is the situation when come to G2. As it enters into the prophase. All right. Now, this cell during anaphase of mitosis. Now, let's take that in metaphase. We have to now take this to metaphase. Metaphase, homologous chromosomes are not going to pair. Homologous chromosomes are not pairing in mitosis. So we can draw these chromosomes. They are not paired. They are not paired. All right. Hope it's clear for you. Hope it is clear for you. All right, people. So I'm drawing it. All right. So it goes like this. What is this phase? This is metaphase. Metaphase. Now you know the spindle fibers. Spindle fibers are going to be arranged like this. Spindle fibers are going to arrange like this. Then you can draw anaphase. Anaphase, what do you do? Just separate these two from one another. So let me show it now. After separation, neither. After separation, these two also separate now. After separation, these two separate. Right? They separate like this. So this is anaphase. This is anaphase. Simple, eh? Simple. Right? This cell during the anaphase of mitosis. No problem. The chromosomes in the gamete formed from this cell, chromosomes of the gamete formed by this cell by meiosis. That's challenging. Right? Now this is for mitosis. Okay, neither. Did you draw mitosis? Haridlamai? Okay, neither. Mitosis, no issue. Right? Mitosis, no problem with mitosis. Now you have to check this, people, what happened during the meiosis. So let me show you the event that happened during meiosis. What happened first? Homologous chromosomes are going to pair. So this is in metaphase. Homologous chromosomes, they pair, neither. Homologous chromosomes, they pair. Homologous chromosomes pair. In this case, these two are a pair. These two are a pair. These two a pair. These two are a pair. They come and align. In the first division, they separate, resulting two cells like this. So this one goes to one cell. This one and this one and this one now goes to one cell. These three goes to the opposite side. Those three goes to opposite side. Those three. Right? In the next step, sister chromatids need to be separated. Neither. Next step, sister chromatids need to be separated. So let's take one of the cell. Let's take both cells. Hope you can see. Yeah, you can see. Right? Yeah, I'll show it like that. Okay? Right? And here, the white color one, come and align here, and then these two and these two. So the spindle fibers are going to attach like this. All right? And then this one, this one, like that. So segregation, next step. Next step is segregation, separation. So this is going to segregate now. So we get 4 cells. Ne? This is going to separate here. So what do you get this side? You get now 4 nuclei forms. This side you get the white color hook, and then the dark color thing, and the light color one. Ne? This side also the same. White color one with the hook, and the dark color one, and the white one. These two, dark color one with the hook, and this one, dark color one with the hook, this one, and this. So you draw one. You can draw different color combination, people, because this is not the only assortment. There are several assortments possible. Depending on that, you can draw your gametes differently. But one of your gamete need to have chromosome with the hook, and then without the hook, and the other one, circle one. Right? These 3 must present in a cell. Is that clear? All right, people. Did you do that properly? Have you understood? Any questions? Okay, ne? Right, done. Look. So, the chromosomes in gamete produced from this cell by meiosis. Now done. No issue. But we are moving to the next question, part B. This is a little difficult question, people, right? Because we don't discuss the theory parts. Asexual reproduction involves only mitosis. Give a reason why offsprings produced by asexual reproduction, if they are different. Now, every time, in asexual reproduction, resulting organisms are identical. But asexual reproduction also can result variation if mutation occur. Right? During DNA replication, if any error occur in the DNA replication, what do you call that? We call it as a mutation. So what are you going to write as the answer here, people? Asexual reproduction involves only mitosis. Give one reason why offsprings produced by asexual reproduction may vary in genotype, again in DNA makeup and phenotype, but not by genotype. Okay, you don't understand that. Genotype means the DNA makeup is different. Why? Right there, people? Due to mutation, Killian. Due to mutation. Now, sometimes, think that people, you live in Kandy. You go to, let's take where? Africa. Or maybe Putlam, little drier area, bright sunlight. You go to, you expose yourself to different environmental conditions. Or maybe you go to Europe. You become little fair, right? Sometimes the environmental factors can influence the features also, right? Sometimes they say phenotype can the visible characters, your appearance is phenotype. Can you write, cut the word and write there, people, phenotype can appearance, right? No DNA change in mitosis. DNA change, if it happened by mutation. No DNA change, but still the appearance is going to be different. Think that you grow a rose plant, right? You find a rose bush, right? Rose stem. You grow one and you give the other one to me, right? So your one, you water it and put fertilizer, look after, remove if any pest person, you remove it. Keep it in a good place, right? Look after the plant very well. After some time, the plant is going to grow well and then bloom flowers. My one, if I throw it somewhere, no one needs to water that. And very difficultly, surviving. And then what will happen after some time? These two plants' appearance. One is green color, flowered. The other one is weakly grown. yellowish, small leaves, no flowers. So that makes the difference. Environment can make the difference. You understand that? So you need to have good environment, right? Good environment, good teachers, neither. Good teachers like me, neither, where you can come and learn. And we tell you the truth every time. We force you to do the questions. Neither. Night time also we take classes for you and then help you with your studies. Those are good teachers, good teachers, right? So you are lucky. Students are lucky to learn from good teachers. Then you bloom, you grow well, you put a lot of leaves, you become greenish color. One day you're going to form beautiful flowers also, right? So that's the environment. So what do you say, people? Here, environment. Right, next one. Part C. The formation of gametes during the sexual reproduction involves meiosis. Give two ways in which meiosis result in genetic variation in the gamete produced. How meiosis result genetic variation? Can you tell me how meiosis result genetic variation? Crossing over, recombination, independent assortment. Very good, right? Crossing over, recombination, independent assortment. Nice. You can write that. Right. Then people, exam question 2014. Did we answer that question? Did we answer that question? 2014 A-level paper question. Did we discuss that? I can remember like discussing it in the class. No, didn't we? No. Yes, we did. We did. Hurry. And then the MCQs. No. Who's that? Rima says no. Not the last page one. Not the last page one. I mean, that's repeating. That's repeating. Right. Then we'll go back for that question I skipped. What's that? 44 chromosome thing we did. 15th question. We did that. There are 44 chromatids present in the beginning of prophase. We discussed that. Question number 14. Lamai. Question number 14. Did I skip any part? You can remember like asking you to do. Or, there are 44 chromatids present in the beginning of the prophase. We did that. No, sir. Part B in the 14th. Part B. Evidence would see the. Part C. Did I do part C? Did I discuss part C? Did I discuss part C? Yes. Hurry. And I'm part B. Right. Now, when you look at a prepared slice through the microscope, right? What evidence tell you that? I told this in the class during the lecture. What evidence tells you that the prophase is longer than the anaphase? What evidence? Right? I told you that in the class, there are a lot of students writing. While writing, one student sneeze. If I fix a camera above to take a photo, this camera is like frequently taking photos over the 4 hours duration of your Saturday class. Frequently taking steps with time intervals. Right? If I take all the steps, what I can see more students writing or sneezing. Sneezing is a quick event. It happens sometimes. Quick event, neither. So writing is a longest event. So when some event is longer, when you prepare a slide, you actually freezes all the cells present in the slide. Hundreds of cells, you freeze them. So they die at their position. Some are dying at the anaphase, some are dying at the metaphase, some are dying at the prophase, some are dying at the interphase. So the longest phase of cell cycle is the interphase. So when you fix a slide, you see most of the cells present in the slide are going to be in the interface. Why? Because it's the longest. Few cells are in the dividing, and out of the dividing, the longest is you can take as a prophase name, prophase and prometa. Shortest is the anaphase. So very few cells, and it's very difficult to spot an anaphase. I think I mentioned you this with the practical also, right? When you observe very few anaphase cells, you can observe because it's the shortest. So as the answer for this question, what evidence would you see on a prepared slide of a bean root tip which would show that the metaphase takes longer than the anaphase? You can see that more cells are present in metaphase than the number of cells present in the anaphase. Right there. Right there. Most of the cells present in metaphase, or you can say that more cells present in metaphase than the cells present in anaphase. This indicates that the metaphase takes long duration than anaphase. In a prepared slide, most of the cells are present in metaphase than anaphase. This tells that the metaphase takes long duration than the anaphase. Right? Than anaphase. Right. What else? How about the multiple choice questions? The second set. Did you discuss? Second set after this page. No? You did? No? No. We said we didn't. No? Okay, we'll discuss them. Which of the following statement is correct regarding meiosis? Happens in prokaryotes. Resulting daughter cells are genetically dissimilar. Homologous chromosome pairing happens in prophase 2. Replication of chromosome happens in prophase 1. Newly formed daughter cells at the end of meiosis 1 have half of the DNA compared to the mother cell. What is your answer? Which of the following is correct regarding meiosis? Happen in prokaryotes. Wrong, neither. Resulting daughter cells are genetically dissimilar. Correct. Homologous chromosomes pairing happen in prophase 2? No. That happened in prophase 1. Eh? Replication of chromosome happen in prophase 1? No. S phase, interphase. Newly formed daughter cells at the end of meiosis 1 have half of the DNA compared to the mother cell. Daughter cells at the end of the meiosis 1, at the end of meiosis 2. Ne? Wrong statement. Meiosis take place in cambium, cortex, pith, leaf bud, anther. What is your answer? What is your answer? Anther. That is correct. Fifth one is the answer. Question number 3. Absent in meiosis. Absent in meiosis. Absent in meiosis. Pairing of heterologous. What do you call heterologous? Different chromosomes. Pairing of heterologous, that never happens. Enang answer is first one. Very good. Huh? There was a segregation of homologous chromosomes. Segregation of homologous chromosomes. Yeah, that happens. Crossing over happens. Synapses happen. Chromosomes become chromatids. Can you correct it as chromosomes become chromatin. Chromatin. Chromatin, not chromatids. Chromatin. All right? Correct it like that. Eh? Question number 4. Most important step in meiosis is separation of chromatids, homologous chromosome pairing and separation, formation of spindle apparatus, dissociation of the nuclear envelope, formation of daughter cells. Most important in meiosis. Tell me your answer now. For this question, this is a tough one, right? Let's see who is going to give the correct answer for this. Most important step in meiosis is separation of chromatids. That happen in mitosis and meiosis both. Homologous chromosome pairing and separation. That is correct. Second is the answer. Very good. Jay Prakash. Good. And next one, formation of spindle apparatus, dissociation of the nuclein, the formation of dot. All are important, but meiosis is about the segregation of the homologous chromosomes. Right? Question number 5. Longest phase of meiosis is prophase 1. Correct. Very good. Question number 6. Migration of chromatids from the equatorial plane to poles occur during. Migration of chromatids from the equatorial plane to poles occur during prophase of meiosis, metaphase of meiosis, anaphase 2 of meiosis. Answer is anaphase 2 of meiosis. Third one is the answer. Question number 7. As a result of meiosis, number of chromosomes double. Haploid cell is formed from diploid cell. 4 nuclei with similar genetic composition result. Diploid cells are produced before fertilization. Daughter cells with higher DNA content than the parental cell are produced. What is your answer as a result of meiosis? What are you going to say? Oh, number of chromosome double, wrong, neither. Haploid cells formed from the diploid cells, that is correct. Second one is the answer. Question number 8. As a result of meiosis, 4 nuclei similar to parental nuclei form. Wrong, neither. They are not similar. 2 nuclei similar to parental nuclei form. 2 nuclei similar to parental nuclei form. No. 4 daughter cells with identical genetic makeup form. No. 4 daughter cells with equal chromosome number. Yes. That is correct. So you take that as the answer. Fourth one. Fourth one. Neither. 2 daughter nuclei with identical genetic makeup result. No. In meiosis, separation of chromatids occur during. You know the answer. Fifth one, anaphase 2. Correct. Question number 10. 2 sets of 6 chromosomes present in 2 opposite poles of a dividing cell with spindle apparatus. 2 sets of 6 chromosomes. 2 sets of 6 chromosomes present in 2 opposite poles of dividing cell. Now, 2 sets of 6 chromosomes means how many chromosomes? How many chromosomes? You answer my question. How many chromosomes? We have taken here an example with 6 chromosomes. 2 sets of 6 can how many chromosomes? 2 sets of 6 chromosomes. How many chromosomes? 12. Good. 12 chromosomes are there. 2 sets of 6 chromosomes are present in 2 opposite poles of a dividing cell with spindle apparatus. Each chromosome has sister chromatid. Each chromosome has sister chromatid like this. So what is this stage, people? Telophase 2 or sorry, telophase 1 or anaphase 1 neither. So we don't have telophase as an answer, right? So what's the answer for that, people? Oh, anaphase 1 neither. Anaphase 1, third one is the answer. Now, if they had given telophase 1, also we can take as an answer, but they have not given that. All right. So only anaphase 1 is given. So without any difficulty, you can choose the right answer, third one, as the right answer. Question number 11. Which of the following is not an essential during meiosis? Not an essential during meiosis. Reduction of nuclear DNA, pairing of homologous chromosomes, exchanging of DNA between homologous, form 4 daughter cells, 2 nuclear divisions. What's the answer? During what they ask people, following is not essential. Exchanging DNA, crossing over is not essential. So the answer is the third one. Crossing over happens, but it's not an essential event to happen every time, right? So sometimes the chromosomes can go without any crossing over. So Mitravaruni, answer එක 3, right? Nadahan. And question number 12. Crossing over take place at prophase 1, metaphase 1, prophase 2, metaphase 2, and interphase. What's the answer? Crossing over take place where, people? Oh, Kianda Balanda occurs in prophase 1. Answer is 1. Very good. Question number 13. Some of the steps of meiosis are shown below. Correct order is shown in. Wait, I didn't read the question even. Wait, I had to read the question first. Don't give the answer before. Right? Form 4 daughter cells, homologous chromosomes separate, exchange of genetic material, replication of chromosomes, pairing of homologous chromosomes. What is the first one? First one is D, replication of chromosomes. Next is E, pairing of homologous chromosomes. Next one is C, exchange of genetic material. That happened after pairing. And then we can take people, the next step is B, homologous chromosomes separate. So the answer is 4. Very good. Question number 14. එනම් මිත්‍රවරුනි කියන්න බලන්න උත්තරේ which of the following statements are incorrect regarding interphase? This is most prominent phase of the cell. Interphase is subdivided into 4 subphases. 4 subphases. Interphase. That is wrong, neither. Answer is second. Second is the answer. That's correct. Right? 15th question is Sira question, right? It's a Sira question. 15th one, only very few people can answer that question properly. Right? Only very few can answer. It is a big question. It is a big question. Right? Can you answer this big question? 15th one. Can you answer the question? Question number 15. If the amount of DNA in a sperm cell of an individual is 3.4 into 10 to the power minus 12 grams, the amount of DNA in the mature liver cell of this individual just before prophase. Right? The amount of DNA in the sperm cell 3.4. Forget about the rest of the part. Therefore, the parental cell 6.8 of the liver cell. But they say liver cell after S phase. Why after S phase? The liver cell goes into the prophase. Right? Then what happened? 6.8 S phase, the content double. 13.6. 13.6. Answer is fifth one. Very good. The Mayali and Jayaprakash. Shaharya. Only two people answered, not Shaharya. Right? Very good. You got the right answer. Question number 16. Others, are you okay with this? Because they don't ask at this level, but they ask after another S phase. We have to multiply it once again by 2. Question number 16. Select the incorrect statement about mitosis. Select the incorrect statement about mitosis. Chromosomes can be observed through light microscope. Chromosomes. Can you see chromosomes through the light microscope? Can you see chromosomes? Answer my question. Answer my question. Can you see chromosomes under light microscope? Yes or no? Eh? Can you see the chromosomes? And can you see yes or no? You can see. We did a practical. Taking anther and then observing chromosomes. What is the chromosome that you use? Sorry. What is the microscope that you use? What is a recommended microscope to use there? Electron microscope or the light microscope? Light microscope, right? So chromosomes can be observed through light microscope. Yes. Chromosomes can be stained with dye. Yes. Homologous chromosome pair. Mitosis? No. Chromosome shorten? Yes. Result genetically similar cells. Yeah. Mitosis? Yes. So answer is third. Answer is third. Moving to next page, question number 17. Question number 17. Which of the following statement regarding eukaryotic cell cycle is correct? Eukaryotic cell cycle is correct. Crossing over take place in metaphase of meiosis 1. Crossing over take place in metaphase of meiosis 1. Crossing over take place in metaphase of meiosis 1. Wrong, not in metaphase. Wrong. Formation of chromatin occurs in the G1 phase. Formation of chromatin occur in G1 phase? No. Chromatin present in G1 phase, correct. Chromatin form from chromosomes in the telophase. No. DNA replication occurs in the G2 phase. No. S phase. Nuclear lump reforms during cytokinesis. Nuclear lump reforms during cytokinesis. Wrong. That happened in the telophase. Formation of mitotic spindle begins in prophase. Formation of the mitotic spindle begins in the prophase. That's okay, neither. Mitotic spindle, yeah, the centrioles separate from one another and then form people. What? The spindle apparatus. That's happening, correct. Question number 18. 4 events of meiosis are given below. 4 events of meiosis are given below. Then which of the following is the correct sequence of the occurrence of the above events? What's your answer? What is your answer? Centrosome move towards the opposite poles, forming spindle. Formation of synaptic complex. Pairing of homologous chromosomes arranged on metaphase. Crossing over. Pairing of homologous chromosomes arranged on the metaphase. No, it's after name. Formation of synaptic complex first. Crossing over second. Centro chromosomes move towards the opposite poles. Centrosomes moves. You can take it as next name. Both, I think both better. Question number 19. 19. Which of the following occur during the telophase 1? Telophase 1 and telophase 2 of meiosis. Telophase 1 and telophase 2 of meiosis. Formation of spindle apparatus by centrosomes. Formation of spindle apparatus by centrosomes. Formation of spindle apparatus by centrosomes. Telophase 1 and telophase 2 of meiosis. Formation of spindle apparatus by centrosome. Wrong, neither. Moving of one chromosome, each homologous pair. Moving of one chromosome of homologous pair towards the opposite pole. Wrong, neither. Decondensation of chromosomes into chromatin. That is correct. Third one is better, ne? Third is better. Stuck, you know. Right, so we'll take third one as the answer. Right, people? Then that's the end of our today's discussion. Right, thank you very much for your participation. And then we will meet you again in our weekend class, right? As in our usual class, right? Thank you very much. We'll have some special program on this, the midweek discussion. We'll set something to revise the previous sections. I'll arrange people a small tutorial for that. Okay. Then thank you very much, and I'll see you. Vanakkam, Aibon, and Salam Alaikum. See you. Good night.